All right, the question that we want to answer here for evolution is what are the three sources of genetic variation within a population? You all know that genetic variation is a requirement in order for evolution to happen. But what causes genetic variation? The first potential cause would be mutations. The second one would be genetic recombination and sexual reproduction. And the third one is lateral gene transfer. We'll look at each one of these three sources of genetic variation right now. The first one will be mutation. You know that the definition for a mutation is a change in the nucleotide sequence of DNA. So because DNA is turned into RNA and then to protein, a change in the nucleotide of DNA would cause a change in the nucleotide of RNA, which in turn may or may not change the amino acid sequence for a protein. If the amino acid sequence does change, the protein might not fold right, and the protein might function differently or not function at all. The result is the mutation could be beneficial, harmful, or have no effect. In order for evolution to happen, a mutation needs to be able to increase an individual's fitness, which means a mutation needs to help the individual survive and reproduce within that specific environment. So this mutation may be beneficial in one environment, and it might not be beneficial in a different environment. So it all depending on how the individual interacts with the environment. The last thing that you need to remember about mutation is that only mutation in germline cells can be passed on to the offspring. A mutation that happens within a cell that's a somatic cell cannot be passed on to the offspring. So if you have a mutation and the mutation increase the individual's fitness, which means this individual is going to be able to survive and reproduce, and this mutation happened in the germline cells of this individual, then this mutation that's beneficial will be able to be passed on to the next generation. And as time goes on, natural selection might be able to select this beneficial mutation in order to make the organism more adapt to the environment, and then evolution happens. So here's a practice for you. Explain how mutations are important in the process of biological evolution. Do this practice on your own. This is, in fact, what we just talked about. And this giraffe is not real. The next method that's able to cause a new variation of a trait or a gene is genetic recombination in sexual reproduction. So if you think about you and your brother and your sister, you might not have the same sex, and you have very many different traits. So sexual reproduction itself is able to produce different genetic variations. So how does that work? First off, there's meiosis, where you have a germ cell, the initial 2N diploid cell, and then after this cell goes through meiosis, you get 4 gametes. It's either a sperm or an egg, but it's a haploid cell that's a gamete. And you will get one of these gametes, which is already different from the original germ cell from one parent, and then another gamete, and those two gametes combine to form another diploid cell, which is a zygote, to produce a new organism. So this whole process selects different chromosomes for the sperm and the egg and recombine them. So this process that's done during meiosis and fertilization, which is right here, is able to produce new genetic combinations, which then can produce new traits, or a new combination of traits, which might be beneficial or harmful or have no effect to the individual. Another way that meiosis is able to help increasing genetic variation is independent assortment. Remember, when meiosis happens, we have many pairs of homologous chromosomes. For a human, we have 23 pairs of homologous chromosomes. And for those 23 pairs of homologous chromosome, each chromosome from each pair can be independently assorted into each one of the gametes. So as this picture is showing, this red and this red don't have to end up in the same cell. You can have a red and a blue end up in the same cell. So this is independent assortment. The process of producing gamete itself give you a unique combination of different chromosomes. So for human, the 23 chromosomes within one gamete could be any combination of 23 chromosomes. So this chromosome is then able to, this gamete is then able to combine with another gamete and then form into a fertilized egg and produce another person. And this person is not gonna look exactly like the parents or any siblings. The next one, this also happens, crossing over also happens during meiosis. Crossing over is when you have two homologous chromosomes, and because those homologous chromosomes are close to each other, one part of the homologous chromosome is able to cross with another one of the homologous chromosome. And then these two parts switch. So as you can see, instead of having all red, you can have a red combined with a part of blue, a blue combined with another part of red, which are different from the initial parental cell. So the parental cell, you only have red or you only have blue. Now you have new recombined chromosomes. So this also increases genetic combination. So here's another practice. Why do sexual reproduction provide more opportunities for genetic variation than asexual reproduction? Try to answer this question. This last one is called lateral gene transfer. Lateral gene transfer is for bacteria. When bacteria has a plasmid, well, many bacteria have plasmid, in fact, but in order for bacteria to survive better, bacteria is able to transfer one of its plasmid, make a copy, and then transfer the plasmid to another bacteria so that another bacteria will be able to receive a plasmid from another bacteria. And the plasmid itself contains chromosome. So as you can see, originally you got two bacteria, one didn't have plasmid, and in the end of lateral gene transfer, this bacteria gets a part of this DNA from another bacteria. We call this bacterial conjugation. About bacterial antibiotic resistance is when if a bacteria has a plasmid that allows this bacteria to be resistant to antibiotic, which is supposed to kill a bacteria, it's able to do bacterial conjugation and send this copy of a chromosome plasmid with the gene that allows this bacteria to be resistant to the antibiotic. It can send this gene to another bacteria so that this other bacteria is able to be resistant to antibiotic as well, even though it didn't develop antibiotic resistance on its own. So we call this lateral gene transfer because this bacteria is receiving a gene from, or multiple genes, from another individual organism, which is another bacteria. So here's another picture showing you how lateral gene transfer or bacterial conjugation works. We have one bacteria that has a plasmid, which is also made out of DNA. And it's able to kind of form a bridge between this bacteria and this other bacteria. And this DNA is going to make a copy of itself and send the other copy into the other bacteria. So now if this bacteria originally is antibiotic resistant, now this bacteria is also antibiotic resistant. This mechanism makes it a lot more likely for more and more of the bacteria to be antibiotic resistant, which means even if a bacteria didn't develop its own antibiotic resistancy, it can make many other bacteria to be resistant to antibiotic, and that is why a lot of antibiotic is not working right now. And scientists propose that there is not going to be any antibiotic that's going to be able to treat bacterial diseases in the years to come, not that long from now. So we'll see. Now, as a review, what are the three sources of genetic variation within a population? We have three things. Think about what they are. All right, we have mutation. This mutation has to be beneficial for this organism's survival and reproduction, and it has to be in the germline. The second one is just meiosis on its own. The meiosis process, the crossing over, the independent assortment of chromosomes during meiosis, and then fertilization, where you combine two gametes. All of these allow new genetic variations to be formed within a population. The third one is called lateral gene transfer, which talks about bacterial conjugation. When one bacteria sends its plasmid, which is made out of DNA, into another bacteria and make it become the genome of another bacteria. This is the end of the video. Hope you learned something, and if you have any questions, feel free to leave a comment or ask me in class tomorrow.